So we're going to talk about the electrifying lives of methane-producing archaea. And these organisms are very small. They are born billions of years ago. They're some of the first organisms on Earth. And they have they're having a metabolism that leads to the production of one of the greenhouse gases on Earth today, which is methane. But methane is not so bad. At that time, it actually warmed up the Earth, which is not really bad news because we like it kind of not too cold. We wouldn't like it very like frozen. And methane is the byproduct of their metabolism, but it has different impacts on our climate and our environment today. It is 36 times more potent than carbon dioxide, which we all know that it is a greenhouse gas. But this one is even more potent than that. So that is a negative thing about the methane that they produce. But it can also be used as a fuel. And I'm going to tell you a little bit about what we can do with it. So it can be used in our economy as a fuel, and it can be used actually by the chemical industry as a building block. So it has quite a few positive aspects as well. It's not just the bad gas. It's also sometimes useful. So who are these organisms that actually produce most of the methane on Earth? They are archaea. And I'm 100% sure that some of you have never heard of them. Maybe I'm not right. But archaea belong to a completely different branch. As you can see, this is where bacteria are. All those little organisms you've heard of, the ones that are pathogenic and they infect us, are belonging to bacteria. But archaea belong to this completely different group of organisms, still small, still tiny, and they are closer to us. They actually are closer to organisms like humans, which is a bit strange, right? So they are not bacteria. And they do something quite different in terms of how they gain energy. And I'm going to go through how cells produce energy, which I know is not something everybody is comfortable with, so I'm going to start from us. In our cells, what we do is we utilize food that we had at lunch, and right now, all of you in your own cells are actually converting that food to carbon dioxide, and you are pretty much scavenging those electrons and sending those electrons to oxygen. And this leads to production of water and carbon dioxide. This is what happens in our cells. Methanogens do something else. They actually utilize similar things, food sources that are very small that existed on the primordial Earth. Very tiny food sources like hydrogen or very small acids. And they actually send those electrons from their food onto another electron acceptor. Something else, not oxygen, because oxygen wasn't there when they were there. So they actually utilize carbon dioxide, for example, and they make methane from this process. This is their byproduct. But I'm going to also tell you about where do they find some unusual electron sources, because some of these methanogens can actually utilize electrons directly. And you may wonder, how would you find that in the environment? Well, there are microorganisms out there that are actually bacteria. They belong to a completely different domain of life. And these bacteria can send electrons from this type of oxidation of food substrates to minerals, to other cells, to the extracellular environment. So when they do so, they can actually also interact with organisms like methanogens. This is what we study. So in my group, we are looking at these unusual interactions between microorganisms. So we're looking more or less at the symbiotic relationship between two groups of two domains of life. And what they do is that one organism is utilizing a food source that it has available, but it has nothing to respire because it's somewhere in an environment where there is no resources. It has no possibility for any other place to put its electrons. So what it does then, it sends electrons out and those electrons are utilized by this methanogen to produce methane. This reaction, this conversion of organic carbon on the account of CO2 would be impossible if these two are not partnered with each other. They need each other. So this reaction is actually completely unfavorable. I'm not going to go into the thermodynamics of it, but I'm telling you it's very unfavorable because it needs to send the electrons out somewhere. And this one has no food. So without food, it cannot proceed. It cannot actually carry out its metabolism. So they need to be together. And I am showing you here one example of such an association. So we've discovered this partnership in 2014. And you can see the red cells are these methanogens, the ones that are producing methane. And in green, we may see somewhere around a lot of the little cells that look kind of like rods, and they're always growing in very tight associations. They grow together, they form aggregates. I'm going to tell you how we study these aggregates and what we learn about them. We have also a very fun video about this interaction where you can see the bacterial organism has some structures on its cell surface that we determined by a lot of very different tools. And it can actually send out its electrons out to the methanogen, which produces methane. But I'm gonna more or less try to bring you through where we can find such organisms. So for example, we went to like a small region close to our university where we sampled the sediment. And here, if I am unsure that you can actually see the blinks. Here, if you look to the little tiny, let's say, setup here, you can actually see that there are quite a few blinks. So this is microbial metabolism in action. In fact, what you're seeing here is bacteria that are more or less attaching to the surface of an electrode at the bottom in the very anoxic region where they do not have access to oxygen. And here, what they're doing, they're sending electrons out and they're producing small amounts of electricity. And we can kind of make a very rough estimate, and we can say that in that area surrounding this electrode, we have about 200 million organisms that can actually donate electrons extracellularly. If we have 10 blinks per minute, so we can kind of make an estimation with that. So what does that mean for you? It means that if we kind of calculate how much energy would that be in terms of surface area. So if we scooped 700 square kilometers of soil from Denmark, and we put it into these little devices, how much energy would we get? We get the energy of a single windmill. They're not making much electricity. It's not going to be solving our energy problems. I promise you that. So this doesn't happen. But it's, however, interesting enough because methanogen can take advantage of this energy that is available from their bacterial partners. So it's good enough for those guys. So how do they do it? How actually do these bacteria and these methanogens send electrons extracellularly, and how can they actually cross the membrane barrier and actually send such charged molecules outside? So what they do is that they will actually transfer those electrons either via cytochromes. So they utilize some molecules that will pretty much be connected or close one to another. So they will pretty much, electrons will pretty much hop from one iron atom to another. Once again, we hear about iron. So iron is very crucial for electron transfer in any type of life forms. So we actually have the same type of electron transfer happening in some of these bacteria. It is just that those molecules are extracellular. They're on the cell surface. And there is another, let's say, more debated theory that we have these pili structures, which are actually having aromatic amino acids that are stacking on top of each other, very much like in certain polymers where people have studied conductivity that is similar to that of a metal. So here you will have a cloud of electrons that will be pretty much allowing for electron conduction through this material. So they have these specialized molecules on the cell surface, and this is how they interact with one another. And the way we study these interactions is pretty much playing detective. So we have to do a lot of things to get the answers, more or less. So some of the things we do is we start with learning about their DNA, so their genomes. We learn about what they express, because the genome cannot really tell us what they are doing when they're on a mineral or when they're interacting with another cell. We learn about their proteins, and we then also actually remove certain genes to see whether those are very crucial for a specific function. So we can really tell, okay, this pili, if we remove the gene to make this structure that is extracellular, what happens? Will they still pair with one another? And we learn that they do not, for example. So all this can give us a lot of information about what happens, what is very crucial for the interaction, what is necessary and what is not necessary. We learn a lot about their behavior in terms of electrochemistry by setting them up on electrodes and figuring out what they actually require under those conditions. And we also look in very fine detail at their elemental composition on the cell surface. So we are trying to identify certain things on the cell surface that may give us hints at how they exchange electrons. With all that information, we can get kind of signatures, information that we can use to go to the environment and actually utilize these tools to really figure out if this type of interactions are very important out there. Are they important for climate? Are they important for methane production in marine sediments, in coastal sediments, in lakes? So this is other type of information we can get. We do both lab experiments with artificial incubations, so we kind of force pair them, we put them into a forced marriage, or incubations that are coming straight from the environment. And the way we sample is by going on these beautiful cruises. For example, this time we were in the Baltic Sea, in the Bornholm Basin. We take meter-long cores, and we can actually slice this cores and get to the region where these organisms are really like living at their best. It's the most anoxic region there. And we can actually determine then what they're doing and how they're doing it. And in 2018, for example, we discovered that a partnership between one bacteria and one archaea, the red one here and the green one is the bacteria, was absolutely dependent on conductive particles. And this is actually a partnership that we more or less figured out from a natural environment. This is from the Baltic Sea, somewhere up north on the Swedish coast. And we pretty much determined that when they have more conductive materials in their environment, they can exchange electrons faster. How and why? That is because they can downregulate the expression of all these molecules that are on the cell surface. They do not need to invest energy in that. They actually then spend all their energy in doing whatever they have to do inside the cell. So it is less energy used for all these extracellular components to be built. All right. And then we also use a lot of tools like, for example, pretty much figuring out who is eating the good stuff. So we can label the substrates and really follow what happens. Who exactly is incorporating the substrate? How are they converting that substrate to methane or carbon dioxide? I'm not going to go into details, but it's very cool, and you can ask more questions afterwards. The purpose of all these experiments is that we want to pretty much figure out what role this type of partnerships have in methane emissions in the environment. So we are interested whether conductive particles that may come from different, let's say, terrestrial environments or rocks, those will actually end up in these marine systems, and they might promote such interactions. And we are curious to know whether we have interactions that are leading to methane emissions that are promoted, or are we actually also able to promote interactions that lead to methane oxidation? So these are questions that we are still addressing. Who is responsible? Will they adapt to environmental changes, to changes that we are inducing on Earth ourselves? So these conductive particles that I'm talking about, it's not just magic dust. It is dust, but it's a little bit, let's say, different. So we have, for example, particles that are released during rock erosion. So when we have a lot of climate extremes, the more climate extremes we're having, the more rocks are going to be kind of damaged through erosion. And these are actually then reaching the marine environment, for example, coastal systems. Industrialization leads to a lot of black soot, like all these black particles that are actually depositing on the ice in the Arctic. And what this means, it means that it's going to melt and it's going to deposit itself on the bottom of our seas. What does that mean to this methane cycling, to methane emissions, to all the processes that happen down there at the bottom of the sea? In agriculture, we use all these chars to promote plant growth because they're quite good. The question is, what does it mean when you have them buried down there? Will we actually promote emissions of methane? And we are using such materials for many other things. We're using it to enhance terrestrial weathering, to do carbon capture, to bioremediate, to enhance ocean alkalinity, and even we actually try to promote ocean fertilization. So we are doing lots of amendments to our ecosystems, and all this can lead to changes that will promote possibly methane emissions. So this is one of the questions that we are trying to ask. But now I told you all these terrible things about methanogens and how bad they are and how many terrible things they can do. There is a good side to them. And I'm going to tell you one single example of the good side of methanogens. In closed ecosystems, we can actually use them to produce a fuel. And this can be, in the future, possibly, one part of our circular economy. So, for example, in 2019, we published a paper where we show methane production only on the account of electricity by a methanogen. What happens is that we grow them in these setups, they grow on electrodes out here, and the only food source for this organism is electrons from an electrode. This is all they get. What happens is that if we have this possibility, we could actually convert the carbon dioxide, for example, from biogas to methane of high purity. What this means is that we can then inject this methane of high purity into the natural gas grid, and then we can actually use it as a fuel. So it is a very, let's say, effective strategy to actually get something useful out of our own waste. Because here, food waste leads to all this biogas. This is not going to disappear. We are going to produce organic waste forever. So using every single bit of that would be quite a smart move. So electromethanogenesis is the good side of methanogens. And all right. So one other thing that I like to tell you about is kind of the questions and the impact that we are trying to address in my team. So we're very interested to figure out how they do what they're doing. We are aware that we don't know how all methanogens out there are actually doing this. And we are trying to find what is their impact on a global scale for sustainable industry and for climate. And with this, I'd like to thank you very much for listening to me tonight. And I'd like to thank a lot of people that have been very crucial, and I'm not even having all of them here, and all the grant agencies that funded my research, and of course the EliteForsk for this award. Thank you.